So we've covered what's available to us now. We keep hearing about a lot of new drugs, especially in the area of immunotherapy. And actually here at the summit, there was an immunotherapy subgroup, and I can't count the number of therapies that they had listed on the slide. It was like 30 or 40 different new therapies that are coming along. So the question is, what are the likely ones to make progress? We were very, very lucky. In 2015, we had 4 new agents approved, which means that here in 2017, we do have those 4 plus the ones that had been approved in 2012 and 2013. And so the new question is, looking in the relapse setting where new drugs are being evaluated, what do we think about what's coming along next? At ASCO, I would say that we, as expected, and I think actually, Mary V, you gave an overview update on those 2 trials yourself, actually, but there were separate presentations about the updates for the DARA VD and the DARA RD, basically confirming what we've seen before in terms of achieving MRD negativity with the combination, which is certainly very exciting in the relapse setting. And then, Joe, you presented the isatuximab, POMDEX, and then very, very early data on the randomized study, and the checkpoint data in combination, some early reports. And so we do have these results coming forward. This is kind of like a boutique observation that actually comes from ASH. I'm not quite sure where this is going with nelfinavir, which is the AIDS drug that was used to overcome bortezomib resistance. This was a small trial from Switzerland that kind of caught people's attention because of the results shown here. There were over 70% of patients did show some benefit with some very deep responses in patients who seem to be bortezomib resistant, but there's been no confirmation of that outside of Switzerland. And so the question is, of the new therapies that are coming along, these were just the ones most recently presented, and we've talked about, yes, we have DARA, which is safe and effective. Of those new ones, which ones will really be sufficiently good to have an added impact? Maybe you could go first, Joe.
Sure. It's tough to be prophetic of the future, but you know, I think you've highlighted a few of them. I would say that the drugs that we'll probably see in the near future in myeloma, 3 that would jump to mind. One is as we've seen it used in other diseases, is the venetoclax,
yes,
as you mentioned, because it seems to have a particular effect in patients who have translocation 11-14 and may have a more broad effect when combined with bortezomib to overcome bortezomib resistance.
Right.
Not sure about the nelfinavir yet, so I'll leave venetoclax in that category. The second drug would be the selinexor drug, which we've used, a brilliant design as a export inhibitor, nuclear export inhibitor, which even in patients who are truly refractory to the 5 big drugs, bortezomib, lenalidomide, carfilzomib, pomalidomide, and DARA, showed a response rate.
Yes, and this is selinexor dex in the STORM trial.
Correct, in the STORM trial. And then lastly, and again, I presented it, of course, but is isatuximab, which is another CD38 monoclonal antibody. Now, daratumab has clearly filled the space.
Yes.
Just this last week.
How much space is left?
Well, there isn't a lot of space. Daratumab just last week was approved in combination with POMDEX.
Exactly.
Yeah.
And so the ISA POMDEX, although we're pursuing this, we're already engaging patients in the study. It may not be the only monoclonal antibody in that space. But the way I look at it, in a disease that we're not curing yet, but we're getting closer to it, of course, the more options we have for patients, the better.
Okay.
There's a few benefits to the isatuximab. It's about half the time of the infusion of DARA.
Well, this is an important point.
Yeah, it is a very big important point for patients. It's a huge point. Our DARA patients, we schedule them for 9 hours their first visit and 5 hours every subsequent one. In ISA, we give the first one under 4 hours and the subsequent ones between 2 and 3 hours. So it's considerably shorter.
Okay.
Now, eventually, we'll probably have daratumab subcutaneous.
But it's taking a little while. But
yeah, it's going to be a couple years away. So I'd say at least I would be watching the path of those 3, of the venetoclax, the selinexor, and the isatuximab.
Do you see any place for the other CD38 monoclonal antibody, more 202?
More 202?
Yeah, good point.
Well, there's more.
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 Yes, play on words.
Yeah.
You love to play on words.
Yeah,
We'll leave
I
this
do.
to you.
Well, I mean, my thought there is similar to what I've said.
Yeah.
More in this space is better for patients because there are patients that have certain side effects of certain drugs and not another. It may influence the cost discussion. If there are more competitors in the market, that's a good thing,
The
frankly.
cost to come
It
down,
could.
Although being the third to the party might be difficult for more 202, but nonetheless, again, I would support an active agent that has activity, that is well tolerated.
Yeah.
We want those drugs available to us. We want more
Even
cards
the
in the deck, as it were.
infusion-related reaction is inferior in comparison with isatuximab and daratumumab, but I don't know if the reason is, again, because more 202 has not been evaluated as single agent, but in combination with Dex.
That's
I
right.
don't know if this
And
is...
then I also heard there's some like mucositis, I think it is, that has a special side effect with the more compound. But
I don't think it's been seen at a high enough number for us to be concerned yet, but obviously time will tell as because the trials are ongoing, and I think we'll probably hear at ASH this year of their most recent studies.
Right. Okay. So it doesn't sound like anything spectacular that I haven't
the
been
other,
aware
of
of.
course, are the checkpoint inhibitors,
Yes.
as we've mentioned. But again, I would just be a bit more cautious in a drug that hasn't shown single-agent activity, that can potentially have some challenges. Although you mentioned well in your pembrolizumab study, you've very well controlled side effects. But we'll see. I mean, I go back to my central point. The more, the better in terms of agents for our patients with myeloma. I just want to make sure we're bringing drugs that are effective, that are going to be safe, that are going to be reasonably, ultimately contributing to the price of the regimens we use. But I'm not sure we're on the verge of seeing a checkpoint inhibitor approved in myeloma. I think those other three we'll probably see sooner than we would.
Yeah, we have to wait to know the results of the phase 3 randomized trial. There is one phase 3 currently ongoing, Pomdex plus minus pembrolizumab. So I think that we have to wait.
Wait for that, yes. Okay, very good. So I thought it would be good to just go a little bit more into detail. Some others, the ones that we haven't talked about so much, we've touched on the checkpoint inhibitors, selinexor, but a lot of discussion coming out of ASCO on the CAR T front, especially with the results of the Chinese not really a compound, but product, I would say. And so the studies are very small. They've shown some patients achieving fantastic response out to maybe a year or maybe 18 months. But really, I'd be interested from both of you to think about, well, BCMA is the main target, but how do you look at CAR T cells for the future in terms of efficacy, and then also any comments about the toxicities?
Well, my thought would be, everybody's excited about it. Patients come all the time asking about it. And I don't want to sound like I'm the dissenter by any means, but I'm still cautious. I mean, realistically, less than 100 patients have been reported.
In total?
In myeloma CAR T cell therapy.
Yes.
Number two, we sometimes seem to have a different toxicity acceptance in CAR T cell therapy. I mean, some of these first patients, out of the first 9,
the cytokine storm,
2 went to the intensive care unit.
This is true.
These people get very sick, and I'm very afraid that we minimize toxicity. It's almost like I feel like when I have my transplant hat on, sometimes as transplanters, we minimize certain side effects.
Yes.
If this were any other drug, people would say, wait a minute, this is not. Thirdly, the response, albeit good, is still not prolonged yet. We need to wait to see, and we have seen a few relapses already.
If you look at the Penn, University of Penn, I think it was 3 out of the 9. There are deep responses, but it's it's a subset of patients even, right?
Correct, absolutely. And then my last point about it is that I'm just very hesitant when people get overly optimistic and start talking about the cure potential. Although
which
the
is the
potential
comments coming out of
might
ASCO.
be there, correct. I mean, that Chinese study was encouraging. It's opening our eyes to it. But this is going to be an incredibly challenging portfolio of treatment to bring to the average myeloma patient. Right now, this is not only in the academic center, this is in very few highly specialized. I mean, I know we're opening one. We have one open now at Mayo Clinic, but it's not easily accessible. And until It's been refined until it's safer, it's more effective, we understand the target better, we understand T-cell manipulation better. I'm a little hesitant to think that it's going prime time. So I don't want to push down anyone's excitement, and the patients watching today, I don't want them to say, oh, Mikhael said CAR T-cell doesn't have a future. CAR T-cell absolutely has a future, but I just think we need to approach it cautiously and take our time to do it right.
So is the Spanish team cautious about the CAR T-cells?
Yes, of course, because so as Joy mentioned, so even the production is not easy, so we don't know how
and the expense on the production, yes.
And so I think that in spite of the CAR T cells, we are going to have a new monoclonal antibodies like the conjugate monoclonal antibodies or the B-specific monoclonal antibodies.
Yes, bispecific.
That I think that they are also very encouraging because probably they are going to have a similar effect to the CAR T cell, but the toxicity is going to be inferior, I would say.
Yes,
so
and much easier across the board, right?
And so we are going to start a new trial with a bispecific monoclonal antibody targeting BCMA with the CD3. So we will see, but I think that these new approaches can be also relevant, and the toxicity will be for sure inferior to the CAR T cell.
Right. So, yeah, so there was some comments about the hype or the hope for CAR C cells. So maybe a little bit of a hype for now, yes?
With some hope.
With some
hope. I would put them both. Instead of saying hype or hope, I'd say hype and hope.
Hype and hope. Hype and hope.
The two together.
All right. And we've really talked a little bit about the same with the checkpoint inhibitors. We need to wait for that phase 3 study, and we've touched on the others for now. Yes.
I guess the only comment I'd add to the bottom of that is that there are new IMiDs. There's two.
Oh yes, the new IMiDs.
Yeah, we have a trial open right now for one of them. Doesn't, as you know, even have a name yet, CC220, which we're being able to use. We've had a few patients on it. So, you know, developing newer versions of the drugs we have now is always beneficial if they're more potent and less toxic. But obviously we need more than just IMiDs in the future, but I'm looking forward to using those next generation IMiDs.
Yeah, but realistically, what strikes me is how much more mileage can we get out of these IMiDs? You know, actually at the beginning of the summit, Leif Bergsegelgård, he presented some of the mechanism of action, and you know, if you move off the target with an IMiD, I mean, how much more can you do with an IMiD? You know, once you've achieved that goal to block the target
I think there's the law of diminishing returns.
Yes,
right. And we've seen that same with lenalidomide and pomalidomide. There's definitely a benefit, but the benefit is getting smaller incrementally.
Yes, I agree. I mean, with the POM versus RAB, there seems to be a better impact on 17p minus maybe. Yes.